From the MIT campus in Cambridge, this is a Pulse podcast. I'm one of your co-hosts, Fang Xie.
And I'm your other co-host, Yosef Bayi. Together we dive deep into biotech stories and feature biotech leaders. This podcast is brought to you by the GSAS Harvard Biotech Club, a trainee-led Harvard biotech organization which is on a mission to bridge the gap between academia and industry.
Yeah, today's guest is James Collins. Jim is a tenure professor of medical engineering and science and professor of biological engineering at MIT. He serves as director at MIT Jameel Clinic. He is also a member of the Harvard MIT Health Science and Technology faculty, a core founding faculty member of the Wyss Institute, and a member of the Broad Institute. Jim is an elected member of all three national academies.
Okay. And then Jim is one of the founders of synthetic biology, and his pioneering research leverages synthetic biology and AI to develop next-generation diagnostics and therapeutics, particularly for infectious diseases such as Ebola, Zika, COVID-19, and antibiotic-resistant bacteria. Jim has received numerous awards and honors, including recognition as a Clarivat Citation Laureate. For our listeners who might not be familiar, citation laureates are considered likely to win the Nobel Prize. Besides his scientific achievements, Jim is also a prolific biotech entrepreneur. The technologies from his lab have been licensed by over 25 biotech, pharma, and medical device companies, and he has co-founded a number of biotech companies. So we can't express how excited we are to have him on today's podcast. Welcome to the show, Jim.
Yeah, well, thanks. Thanks for having me on your show, and welcome to my office.
Yeah, thanks. Yeah, there's a lot of cool books up here, so we've been admiring those.
Yeah.
Welcome, Jim. And we always like to start our podcast getting to learn a little bit more about our guests. So could you tell us a bit about your upbringing and what led you to be interested in pursuing a scientific career early in your life?
Yeah, I was brought up in a very technical family. So my dad, who's still alive, was an electrical engineer who worked in the aviation and aerospace industry, and my mom was a mathematician and mathematics teacher. Many of my uncles were engineers. And my dad, in particular, would bring a lot of stuff home from his different projects, exposing us to all different cool tech, and actually even set up an electronics lab in the basement. And, notably, his team helped develop one of the optical guiding instruments for the Apollo 11 mission, posted behind here. Wow. And so I was 4 years old watching in the house when they landed on the moon. And so it was a very proud day in the house. And notably, there was a Collins on that mission who was actually stuck in the lunar orbiter, Michael Collins, who didn't get to go onto the moon. But, it was interesting that, so I was exposed early on, and my dad would show me cool stuff being shot into the sky and shot out of the sky. And when I was young, his dad lost his vision, to glaucoma and other conditions. And then a few years later, my mom's dad had a series of strokes where he became hemiplegic, losing control and sensation on half of his body. And I was really struck again by all this cool tech that my dad and his colleagues were developing, and really nothing being done for these two guys who I cared very much for, and became interested at that stage. And could I begin to develop technology that could restore biological function in humans? And this was in the 70s, really before biomedical engineering was a discipline. Became also intrigued on to what extent could I use that to improve my athletic ability. I was an athlete, but I wasn't as good as I'd like to be, and I think no athlete is as good as they want to be. And I thought maybe my interest in science could actually help. Then I think it helped a bit. But it actually, those really formative aspects of my family helped really encourage me to get into science and engineering.
Interesting. Yeah. You were accepted to MIT and Holy Cross, but eventually you attended the Holy Cross as the senior in high school. So how did you make that decision? Tell us.
Yeah, so it's interesting. Firstly, Holy Cross is a fantastic liberal arts college in Worcester, Mass. And it was interesting. So I had been intrigued by the possibility of really wanting to study biomedical engineering. But at the time, it really wasn't yet a discipline. And so I then really went for the area closest to it, which was electrical engineering. So I began to apply to a number of colleges, universities in electrical engineering, including MIT, as well as WPI, so Worcester Polytechnic Institute. I remember visiting the campus, and my dad and I were going around, and there were jet engines in the hallway, and it was smelling of oil, and very serious, excellent engineering school, but it really didn't have the feel that I was looking for. And I had been fortunate to win the Holy Cross Book Prize, which was given out to juniors in high school who were engaged in their communities the year before. And Holy Cross in that day then would send in the mail a lot of brochures about the college. And I was aware of the college also because they had a very good summer basketball camp that many of my friends had gone to. And so I said to my dad, hey, can we just go across the city while we're down here to check out Holy Cross? And we went there, and in contrast to the engineering schools at the time that I visited, two key things. One is that there were a lot of girls, a lot of female students, and they were smiling and looking like they were having a good time. And I then went through the process further, and really, as you said, came down to MIT and versus Holy Cross, and in the end chose Holy Cross. And I think it was one of the best decisions that I made in my life for a few reasons of why I made it and then why it was so important. You know, I at MIT in 1982, 83, when I was coming out of high school, didn't have biomedical engineering yet as an undergrad program. Was largely a male campus, and really wasn't focused on innovation the way it is today. It was focused on training professional engineers to do really solid engineering. I had gone to an all-boys Catholic high school, Bishop Gurton High School in Nashua, New Hampshire, which is an excellent school, but wanted to have a co-ed experience. Okay, MIT was co-ed, but not as co-ed as Holy Cross. I also thought Holy Cross was going to give me a very broad education in the classroom. MIT would give me a great educational classroom, but I thought I would have a better experience outside the classroom at Holy Cross than I would at MIT. And I made the decision to then go to Holy Cross, and I ended up studying physics. And it was interesting. My parents were very supportive. My dad was a little puzzled. My teachers weren't as supportive. They thought I really should commit to MIT, which was a higher ranked school and different, very different school. But notably, I met my future wife early on at Holy Cross. In fact, my first year calculus class, and she had made the choice of Holy Cross over Harvard. So we maybe were one of the only two students to have made those kind of choices at that time and to this day. And, we really, I think, lucked out by making that. And I look back, my time at Holy Cross was fantastic. I had so many engaged professors who really took interest in me, helped develop me as a scholar, as an intellect, many of whom I'm still friendly with today. And I got a broad liberal arts education, both in the sciences and outside the sciences. So not only was I exposed to fundamentals, physics, mathematics, chemistry, biology, but also was trained in not only critical thinking, but how to communicate across disciplines and to non-scientists, which has actually served me well because I've changed fields multiple times as a young professional.
It's a good point. I think in college, that's kind of your last time to get a very broad education compared to later when you go to grad school. And then did you, we know you got into doing scientific research while you were an undergrad at Holy Cross. Could you tell us how you got into that and what kind of led to your interest?
Yeah, it's interesting, again, for young folks, and I know you have more grad school trainees, but I like to talk to the undergrads, and I teach undergrads here at MIT, is that, rarely is your path linear. Rarely is it deterministic. It's often scattered and quite stochastic, and mine was as well. When I first entered Holy Cross, I knew I was going to do physics, but I was also quite interested in business and thought I'd be a businessman. At the time, there was a book called In Pursuit of Excellence that was written by consultants out of McKinsey, I believe, that highlighted a number of entrepreneurs, including Hewlett-Packard. And I was really taken with that, and actually launched that next summer with two of my friends from high school, Michael Malley and Rob Prunier, an underage dance club in Nashua, New Hampshire, that was for those who were under drinking age, but you could go and have a great time. And we had a great time with it. Lost all of our money, but actually had an interesting time as young entrepreneurs. Realized that really wasn't where I was going to go. My sophomore year, I considered medical school and became pre-med unofficially, taking orgo as an overload class. Had the opportunity to shadow doctors at UMass Medical Center in Worcester, and realized that I didn't want to interact with patients. I didn't like seeing stuff coming out of people's bodies. So I crushed that out. Junior year, I became intrigued by policy. Could I be a policy person? So this is now about 1985, 1986, which in the U.S., our dominant worry then was nuclear war and mutually assured destruction. And from the science side, you had a number of folks, largely on the right, advising about the value of nuclear weapons, and you also had a push for the Strategic Defense Initiative, SDI, Star Wars. Could you have a high-tech defense system against missiles coming from the Soviet Union? And I, again, got intrigued and dug in a bit, but realized I didn't think the policy folks were having much of an impact, and it really wasn't where I thought I could match where my interests were, and got taken a bit with research, and was fortunate enough to be selected for what was called the Fenwick Scholar program at Holy Cross that allows you to do a full year of research effectively in place of classes. I did take classes on top of them, and had an opportunity to work on functional neuromuscular stimulation. That you could deliver electrical signals to stimulate muscles in paralyzed individuals to produce functional movement. And I had a marvelous biology professor, Ken Presswich, who took me on. This was outside his area, but he nonetheless took me on. And I had additional input from John Axland in psychology and Ed Kennedy in physics, as well as Peter Grigg at UMass Medical Center, and got exposed to these amazing minds and amazing mentors, and got really excited about doing research, which led me to consider and go to grad school. And interestingly, now as a senior full-time professor, I actually am involved in business. I'm involved with medical docs, married to medical docs. I'm involved in policy, but I primarily do research and teaching.
Yeah. That's a diverse background. Yeah.
It's all circular. All those like areas you explored as an undergrad kind of come back now.
They come back now, in different ways. And I think being a professor, it's a marvelous profession given the flexibility we have, the opportunity to really try to discover new things, but also utilize and leverage new discoveries for applications. In addition to training young folks who are always so exciting to interact with.
And after Holy Cross, you are actually awarded by the Rhodes Scholarship and allow you to continue the PhD study at Oxford.
That's right.
Yeah. And there, the research, what you're doing is the mechanic, right? Mechanic.
Yeah, so I was very fortunate to be selected as a Rhodes Scholar and chose to accept it. And I say that chose because I was being advised by many of my scientific mentors not to take it for two reasons. One is that Oxford was not the place to do science in the UK, University of Cambridge was. And second was not to do science, graduate work outside of the U.S., stay within the U.S. system. I ignored both bits of advice, and I'm really happy I did, because I had probably my most meaningful intellectual experience of my career as a graduate student at the University of Oxford, primarily for the experience in the Middle Common Room, which is kind of the graduate student hangout, where I got to hang out with dozens of individuals across multiple disciplines every day for hours on end. But my research there was really twofold. I focused on biomechanics of human locomotion and sports mechanics. And I got engaged in neurocontrol and nonlinear dynamics. So as a grad student, I had the really great fortune of beginning to collaborate with Ian Stewart, who was is a math professor at University of Warwick. And the brief anecdote there was I was working at the Oxford Orthopedic Engineering Center as part of the University of Oxford, and Ian had published a book review in The New Scientist in which he had reviewed a book called Natural Computation. And this book at one point was talking about animal locomotion and robotics based on animal locomotion. And Ian at the time was studying coupled nonlinear oscillators. And he, with Marty Golubitsky, was studying symmetric such networks and had arrived at interesting mathematics for how three coupled nonlinear oscillators would interact and produce interesting patterns. And in his review, he speculated, is anybody interested in studying a three-legged cat, because I have a model for how they work. And I gave him a call, and I said, I'm not interested in three-legged cats, but I am interested in four-legged cats and other legged animals, and I would love the opportunity to talk to you. I think we could do something together. And he invited me out to visit him at University of Warwick, which is about an hour train ride. And we struck up what became a 10-year collaboration. Wow. From that, which actually pulled me into the world of nonlinear dynamics, dynamical systems, and chaos theory, which was Ian's area of expertise. And it launched me into a whole decade of research.
That was really cool. And you had done this work on muscular research again, and after that, after doing your PhD, you ended up starting your lab at BU, focusing on synthetic biology. So we're wondering kind of what went into your decision making to explore a new area when you're starting your lab, and also what was the experience starting a lab after your PhD, where you're going from being advised, being independent on your own, to advising people to be independent.
Yeah, so I was fortunate to get hired by Boston University right out of grad school. So this is 1990. Postdocs were not yet kind of the expected de rigueur in biomedical engineering. It was beginning to become a trend, but not yet fully expected or required. And so went right in as a research faculty to start. Notably, I did not work on synthetic biology for the first decade. Didn't exist yet as a field, but instead was working on applying nonlinear dynamics to problems in physiology and medicine. And notably, we looked at three dominant streams. One was human balance control. Can we study how people maintain their balance to better understand the underlying mechanisms and improve balance? Second was, can we understand sensory function, both touch sensation and proprioception and how it impacts motor control and how we could enhance both. And third was cardiac function, and did that from a nonlinear dynamic standpoint. I can speak to the transition to synthetic biology later. But, the challenge when one starts a lab is multifold. You're going from just focusing on yourself to now you have to direct young folks. So you've got the challenge of really matching that project that's right for that student, both from their interest and their background, what else they need to be developed on. And that's non-trivial of how to do that. And as I tell young people now, I'm now 35 years a professor, so I've been doing this, is that for the first few years, you're so much more productive than anybody who you're directing. And you need to keep that in mind because productivity is still important. So you have to be true and honor the commitment you make to that young person, but you also need to advance your research program. And so you're still your most valuable member for those first few years as you begin to develop and begin to now establish your lab.
Oh, wow. Okay. So first couple of years still in a lab while you're also advising some of your students?
That's right.
Yep.
Okay. And then, so you mentioned 10 years more on this nonlinear dynamics, and then what led to the shift to synthetic biology?
So the shift into synthetic biology was interesting. So I had no background in molecular biology. I had never taken a class in molecular biology. And interestingly, molecular biology did not have a presence in biomedical engineering. There were interesting debates, political debates within our discipline through much of the 90s that the term genetic engineering had been misappropriated by the molecular biologists, that there really wasn't much engineering to genetic engineering. It was mainly molecular biology. But what was interesting is that Boston University had leaders who had backgrounds in molecular biology. So the chair of the Department of Biomedical Engineering in the mid-90s was Charles Cantor, who had been one of the PIs of the Berkeley effort as part of the Genome Project. Charles DeLisi was our Dean of Engineering, and he had been the young program officer at the Department of Energy who had conceived the Genome Project in the 1980s. To the young folks listening, the dominant project in the American science scene in the mid-90s was the Human Genome Project. Every other week on the cover of Nature Science, it was something about the Genome Project. And interestingly, biomedical engineering had not played a big role in the project. Bioinformaticists certainly did for computation of how do you put together these sequences, but biomedical engineering, no. And biomedical engineering as a discipline had basically stopped at effectively tissue engineering and maybe a bit cell mechanics. Curriculum and research programs did not go really to cell dynamics or to molecular level. So we were kind of flying blind. And in about 1996, the two Charleses began to introduce me to folks in the Genome Project, Eric Lander, Lee Hood, and they began to share with me their excitement around these parts lists that were emerging from the genome effort, and that they wanted somebody like me who had come out of nonlinear dynamics control theory. Could I begin to work on these parts lists to figure out how they fit into networks inside living cells? So it was really the early days of systems biology, which is a field that Lee Hood really pioneered. But with my students, we began to look and realize that the data weren't really available to do reverse engineering meaningfully. And so we ran away from that problem. And as I like to say, that you have two kinds of kids who become engineers. You have the kids that take apart a radio to see how it works. They basically become systems biologists. Rarely do they put it back together. But the more common kind of future engineer is the kid's a tinkerer. The kid that goes in your garage, goes in your basement, goes in your attic, and makes a go-kart out of a couple 2x4s and wheels off of a baby carriage. And so they forward engineer stuff out of tinkering. And so with Tim Gardner, he and I began to think, well, instead of reverse engineering natural networks, could we build networks from the bottom up? And this is what led us to help launch what became synthetic biology. And the notion was, could you make a small circuit out of molecular parts that functioned as designed, that had interesting functionality? And part of our motivation to actually build, in this case, a toggle switch, was could we use it to test our mathematical models? So I had come out of, in part, the math biology community, both as a grad student and early faculty, and this was a marvelous community then and marvelous community today. But in the early days when I was part of it, and it kind of took off in the 1960s, 70s, I got engaged in the late 80s, it was a community where the model was the end goal. And it was really trying to make the model as strong as possible, and you would at best try to see if it matched a published data line or plot in a publication. You never really had interplay between computer experiments and the computational or the analytical work. That began to change in the early 90s, primarily in the neuroscience field. So folks like my BU colleague Nancy Capel was working with Avis Cohen and Eve Martyr on looking at small isolated neuronal systems from the somatic gastric ganglion, from lamprey spinal cord, where they could test predictions from mathematical models experimentally. Tim and I got intrigued, could we do something like that inside a living cell at the level of genes? And based on mathematical modeling for the toggle switch, built this inside E. coli, and it actually functioned as the model had predicted, helping to validate the model, but also helping set the stage for a new biotech field.
For our listener, that's the paper you're talking about, actually your 2001 for E. coli, right? That paper.
So 2000 in Nature.
Yeah, the very first paper, and the thought as one of the landmark paper for genetic circuits.
That's right.
Yeah.
It was interesting also from, there was stuff in the air at the time, in that our piece was published in January 2000, and alongside it was a paper from Mike Ilowitz and Stan Liebler at Princeton. And so Michael and Stan, working completely independently of us, we were not aware of what they were doing, they were not aware of what we were doing. We're two physicists working in the physics department and the molecular biology department at Princeton, and had similar notions to us. They said, could we build circuits inside E. coli, motivated by modeling from nonlinear dynamical systems? And they actually used the same three inducible repressor systems that we used. We did two genes that were mutually repressive. A was trying to shut down B, B was trying to shut down A, that could lead to a bistable genetic toggle switch. They instead took the three repressors and set them up in a cyclic ring. A inhibited B, B inhibited C, C inhibited A, to produce an oscillating circuit called a repressolator. So these two papers that were published back to back ended up serving to launch what became the field of synthetic biology.
Back then, about that time, you just like going and put your energy into synthetic biology. But I guess because of your background, and you mentioned you have a very good collaborator and help you, but for a new lab, and you definitely need some resources and a little bit of less risky project to make the lab survive. Could you tell the audience why you decided to pursue that direction, synthetic biology?
It's an interesting question. I'll make a couple comments then for advice for young folks. I think, we didn't intend to help launch a field. We intended to work on an interesting problem. And we saw there was an opportunity. And, again, for the young folks, your career is not going to be linear. It's not going to be deterministic. It's going to be nonlinear. It's going to be stochastic.
Yeah.
And mine has been and continues to be. And we were encouraged by the two Charleses, Charles Cantor and Charles Halicin, to begin to explore how we could enter this field of molecular biology, and saw there were really interesting problems, and that we could enter with our approaches and our interests. And it turned out to be marvelous. So we made the shift. So I didn't change my whole lab overnight. Okay. Tim was willing to come in. Charles Halicin gave him a fellowship. Charles Cantor gave us some lab space. So it started with really one project that then began to grow, and we could convince more and more outside groups to give us funding. I could gradually bring on more and more young folks to take on these projects, and it was a gradual transition. So a few points. One is that I think as people think about, how do you enter a new field, I always say that you want to obviously go into areas that you're interested in and pivot from strength, so that you want to, and know where your weaknesses are. So by pivot from strength, it's bringing a new perspective, a new set of tools, new approaches that you're expert in into an area that maybe they don't have. In this case, it was an engineering concept, engineering principles, computational modeling for design into molecular biology. Second is find collaborators that can help you. In our case, it was Charles Cantor, one of the best biotech and molecular biologists around. Second is when, talking about, the risk, that it's a challenge, right? You should be willing to take risks with your career, but you need to do it smartly. And as young people are building out their programs, if a key thing that I like to recommend is, the art of the really successful academic is to find the low risk, high gain projects. Meaning you don't ever want to go after the low gain project. Why would you do that? That's boring. Don't waste your time. You can have some high risk, high gain projects, but you don't want to only have the high risk, high gain projects. You're not going to be able to have the productivity to get to the place to do it. So you need to have that mix. And if you can identify the low risk products for which you have an unfair advantage from a time, from a resource, where you can get to those discoveries, those insights relatively quickly in a competitive space, find that niche and build that out. And then, you want to have an interesting portfolio of projects for yourself and for your trainees, where it can be a mix of the really big breakthrough type, and then a mix of the follow-ons to go deeper and build out. And so you want to have that smart portfolio as you're building out. And okay, you have Andrew Wiles that squirreled himself away for 7 years in Princeton to solve Fermat's last theorem. And it's rare that you can successfully do that. And it's better instead to have that mix balancing risk and gain.
Yeah, that's good advice. Yeah, and I think what something you said in the beginning is like not really looking to do something exciting, more of like finding the problem or like the lack of knowledge and exploring that, because that's something that there's promise in it, and it's going to be exciting if you discover it.
Yeah,
that's right. That's right. And again, I know there's trainees, and maybe some of them are just starting their training. We have here at MIT or grad students just starting, and the advice I give to grad students is make sure you take on a project that satisfies 3 criteria. 1, it's exciting to you. 2, it's exciting to your advisor, and 3, it's exciting to the outside world. If you don't hit all 3, don't do the project.
Oh, wow. Okay.
Okay, great.
Yeah, that's really exciting to hear. And you mentioned like kind of like the early days and like switching things up as your lab's grown. And at some point, your lab's kind of switched or kind of focused more on like diagnostics and like stuff related to infectious disease as well. So I was wondering how you got into that, like the diagnostics for viruses as well as like other kind of infectious diseases.
Yeah, so it's interesting. So an earlier shift that was related as well is that about 2 years after we did the toggle switch, which had been done in E. coli, we also got intrigued, could we then turn back to systems biology? Now I say turn back in that we had not done anything in systems biology. We had actually helped launch synthetic biology more or less as a negative reaction to the encouragement to get involved in systems biology. But we then became intrigued, could we use the synthetic biology tools we were developing to begin to help tease out the complexity of natural networks?
Yep.
And we naturally gravitated toward doing this in bacteria, including E. coli. And with some success, where we could better understand how these natural networks are wired together and how they dictated the response of these microbes to different drugs and other perturbations. In the bacterial world, the drugs are antibiotics. And so we helped launch our interest in antibiotics research from a systems biology standpoint. That pulled us into the infectious disease world. And one of the challenges in antibiotics is, can one get after narrow spectrum antibiotics, those that only go after a particular pathogen and don't go after commensals or other pathogens. But the challenge with the development and use of such narrow spectrum antibiotics is, do I know what the patient has? So you need new diagnostics. And so we became intrigued, to what extent could we use synthetic biology to both program microbes to serve as living diagnostics, as well as to create a whole new suite of programmable sensors that could respond to RNA or DNA from a pathogen. We began playing with these sensors. Alex Green, who was a postdoc, co-supervised myself, and Peng Yin, developed a so-called toehold sensor. This was a mRNA in bacteria that were designed to be repressed in an off state that would then flip on from a translation standpoint in the presence of particular RNA. We then realized, with an advance by Keith Pardee, another postdoc at the time in my lab, that you could use these in the context of cell-free systems that had been freeze-dried onto paper.
Yeah.
And that you could freeze-dry cell-free extracts along with synthetic biology constructs that then, when rehydrated later, became alive as if they were in a petri dish, test tube, or in a living cell.
Yep.
And it opened up this possibility of paper-based diagnostics that could be produced for pennies for each sensor, but could be freeze-dried and thus distributed without need for refrigeration around the globe. You stick it in your pocket, get on a plane, take out, come on. And so you could address challenges of detecting infectious diseases in low-resource settings. And we initially did this for antibiotic-resistant infections. We then did it for Ebola in the midst of the Ebola crisis of 2014. And we did it in the midst of the Zika crisis in 2016. And the last crisis, our systems were actually deployed as research tools and surveillance tools in 6 different countries.
Yeah.
It had an impact on that outbreak, which we were very proud of.
Yeah.
That's really good to hear. And then a lot of these like diagnostics we used when the COVID pandemic hit to like adapt to that. So could you tell us a little bit more about how you got involved with that? Because I feel like at that time there was like so much science being thrown in to like help out with COVID, and kind of what were you thinking in terms of?
So it was interesting. I mean, you think back now, it's a little over five and a half years ago when that all hit, and a few things to note. One is I think the U.S. and broadly the world didn't handle the diagnostic challenges well in the beginning. I remember they were restricting labs to sending all samples, I think, to the CDC who had the wrong PCR primers. And so we were missing a lot of schemes. Anyways, we had advanced our synthetic biology efforts and CRISPR-based efforts academically, but also as part of a startup company called Sherlock Biosciences. And at the time, also, academic labs could stay open provided they were working on COVID-related projects. And we did, primarily from a diagnostic standpoint. And working with Sherlock, starting in late February, we repurposed our efforts to see could we use our synthetic biology and CRISPR-based platforms to come up with a CLIA lab-based diagnostic. And we got excellent data together, and by early May, had actually had FDA approval. So it became the first FDA-approved CRISPR synthetic biology product that was a COVID-based test. Teamed up with a group to manufacture upwards of, I think it was a million per week. And notably, we at the company didn't think you should benefit or profit from the pandemic. And so we set up a nonprofit foundation, the 221B Foundation, named after Sherlock Holmes' fictional address on Baker Street in London, where we made available all of the IP and the tech around our COVID test. And any group could use it. With the expectation they would give the profits back to the foundation, which would then use it for STEM efforts. For underrepresented minorities. And the company, Sherlock itself, also provided all of the profits from our COVID tests to the foundation. And 5 different global diagnostic firms actually adopted the test, as did Nepal as one of their national standards. And at the peak, I think there were 10 million tests per year being run on these platforms, which was really satisfying that we had a part in trying to deal with that horrible pandemic.
That's really good. It's like helping out with the pandemic, but also the fact it's kind of like a nonprofit avenue and like renewing the funding for STEM, the future scientists.
You lab exploring the multiple directions. And one is very exciting field is leverage AI with synthetic biology to identify, even design new antibiotics to fight off antibiotics resistance bacteria or superbug, what we call it. The recent achievement is Halicin. Could you tell us about that, the story behind the Halicin?
Happy to do. So, as alluded earlier on the podcast, and going back now about 20 years ago, we got interested from a systems biology standpoint to use network biology and machine learning in the context of network inference to better understand how antibiotics act and how resistance arises, really with the goal of boosting existing antibiotics through adjuvant inputs. And so work with Mike Wahansky, Dan Dwyer, Graham Walker, my colleague here at MIT, Mike Lobritz, who's now a leader at Roche. We uncovered that antibiotics act more complicated than we had thought, where it's not simply inhibiting a target. There's actually a downstream reaction from the microbe that contributes to the efficacy of the antibiotic, where you're triggering stress responses that have metabolic demands that lead to toxic metabolic byproducts that can damage different aspects of the bacterial cell, DNA, protein, membrane, that then trigger additional stress responses that lead to additional metabolic demands, lead to additional toxic metabolic byproducts, leading to this cycle of damage that contributes to the killing that results from antibiotics. That was our initial kind of body of work. Now, fast forward to the late teens, 20 teens. Here at MIT, there was a new effort, new institute-wide effort focused on AI that was being announced in March of 2018. And we had a symposium to mark that launching, and I was invited to speak, as were many other faculty, including Regina Barzilay, who's one of our star AI faculty here at MIT. We sat next to each other and realized we both had interest in AI and drug discovery. And I share with Regina specifically our interest in antibiotics, and she was intrigued. And we struck up a collaboration and brought on also Tommy Jockola, another MIT AI faculty member, and put together a project that was effectively unfunded to start to see could we use deep learning to discover new antibiotics. And we put together a very small training library, applied to E. coli, trained deep graph neural nets, applied them to various libraries of compounds, and actually uncovered Halicin, named in homage to Hal, the AI killing system from 2001: Space Odyssey, that in the movie killed humans, our Halicin kills bacteria. And in fact, Halicin's a remarkably effective new antibiotic, acts by new mechanisms of action across the membrane and is remarkably effective against multi-drug resistant bacterial pathogens. That led us to launch what's become now the Antibiotics AI project here at MIT, with support from the Audacious project through TED, as well as Defense Threat Reduction Agency, and now ARPA-H, major effort. And we're working closely with a nonprofit we launched called Phare Bio, which is designed, set up to basically take the most promising molecules from our effort to the clinic. And we very recently showed you could use AI not only as a discovery tool, but as a design tool. So we now have been using generative AI. So many of the listeners of the podcast have used generative AI to create text or to create images. We use it to create new antibiotics. And we were able to create never before seen antibiotics that act by new means of action, effective both against MRSA, so methicillin-resistant Staph aureus, as well as multi-drug resistant gonorrhea. And we just published this in Cell last month and got a really nice, strong, positive reaction from the world community, who I hope will build upon this excitement to help advance programs to address the AMR crisis.
Yeah, it's a really good point, because I feel like that's an issue of like developing new antibiotics. I feel like AI is going to be very helpful to efficiently do it.
That's right. Yeah, I think we and others have shown AI can do a really good job in the early discovery. I think we have work to do to improve further down the developmental pipeline. So using AI to get after optimization, ADMET, PKPD, and even the design of the clinical trials. And I'm hopeful that AI will help. It's a tool. It's nothing magical. It's, I think, a useful tool that's going to be used increasingly. But we also need better engagement of the world community to fund those later stage trials versus, right now I think we have pretty good support and buy-in on the preclinical discovery and development efforts. But we need to make that next leap to actually advance these to the clinic.
During the COVID time, you also got an interview and talk about use the same platform to search for like antiviral medicine for COVID. Tell us about that.
So there, during the pandemic, working with Wengang Jin, who was then a postdoc here at MIT, he's now a professor at Northeastern University, has remained a very close collaborator, and was one of the key scientists on our generative AI project, which had been led by Adi Krishnan, Melis Anatar, and Jackie Valeri. Wengang Jin was key for one of our generative efforts, led an effort to really get after combinations therapies for COVID. So one of the challenges, can you do repurposing, but then also from repurposing, can you get after interesting combination therapies? And we made some really nice progress there. We're now extending what we're doing on Halicin to broadly viral discovery and now viral design, and also working with Felix Wang, a postdoc in my lab, and one of the founders of a company called Integrated Biosciences on the West Coast that he founded with Max Wilson, who's a faculty at UCSB. They actually showed you could use optogenetics to virtually stimulate the integrated stress response as a way to uncover new modulators of the integrated stress response, and in doing so, identified small molecules that are actually broad-spectrum antiviral. So I think this intersection of AI and synthetic biology broadly is opening up a lot of possibilities for new treatments against infectious diseases.
That's really good to hear. And a lot of the work we talked about is using synthetic biology to study bacteria, virus, and we want to know a little bit about how you're thinking to use it for mammalian cells. Could you share your thoughts on how you could apply synthetic biology for use in human cells?
Yeah, so we've done, going back, I had a brilliant grad student, Tara Deans, who developed one of the first circuits of mammalian cells that was a very tight, tunable system that involved both repressor proteins and RNAi, and could have effectively a true off and then could do a very high on, and opened up possibilities for nice control for gene and cell therapy. Building on that, it was working with Wilson Wong, professor at BU, on what became basically super CAR T therapy. So using logic gates to create smart CAR T therapies that could sense multiple antigens on the surface and make decisions about which cells to attack on the tumor side. This, in part, led to the founding of Senti Biosciences, which is a public biotech company led by one of my former students, Tim Liu. And Tim and team have actually advanced logic-gated CAR NK cells for treating various forms of cancer, AML in particular. And they've actually reported very promising positive results. So I think the notion that one can program cells with decision-making capabilities and the ability then to produce therapeutics in a very highly specific control way, I think can be transformative in many different therapeutic areas. And Tim's company in particular is already showing great promise.
Okay. But any specific challenges, do you think?
I think there's always challenges, and that biology is not yet an engineering discipline.
Yeah.
And we really brought up on inorganic substrates for engineering for the most part. Biological systems are very small, very dense, stochastic. They evolve. They themselves change up. And so it's very difficult to get a good handle on the complexity of the biological systems. I do think that bioengineering, synthetic biology, and AI together can help us make advances, but we have a lot of work to do. So in the end, I think it's probably the stochasticity, the uncertainty, and the flexibility of the biological systems can get in the way of some of our best our engineering design.
We briefly talked a little bit about, your choice to kind of pivot when starting your lab, and you gave a little bit of advice on what people could do to start their labs. I wonder if you could suggest anything. I know you kind of mentioned choosing something you're excited about, or also just finding a problem that's unidentified. But would you want to expand a little bit about that?
Yeah, I'll maybe restate some advice I got as an early faculty member from Charles DeLisi. I'd had a proclivity for getting involved into some messy situations, and he said, Jim, you're wasting your time on a lot of that. He said, really what you need to do is put your head down and focus on doing exciting research. Exciting research is exciting to you, exciting to others in your group, and exciting outside. And it's advice I shared earlier in this podcast, and I really took to heart. And I think for young folks, it's focus on doing good science. There's so many distractions in this world, and I think it's important to focus on doing good science. And I think too often people focus on the secondary, the tertiary goals. I think you obviously want to find something you love, realize that most of your good ideas aren't going to work, so you got to develop a lot of good ideas. Biology in particular doesn't really yield its secrets very nicely. And in this office, the number of times that we think A should go to B should go to C, that don't go to B or C when you get into the lab is stunning. And so I think you want to make sure that you're resilient and are set up with a supportive group of folks in your lab and colleagues and friends to deal with. And I think maybe specifically then to the young folks coming up, and I think the strong advice I'd give there is to shut off your phones as much as possible. Read the literature as much as possible. Really commit to reading the literature and being an expert. And it can seem overwhelming, but you got to start somewhere and make that commitment. I think it's then talking widely about your ideas and about what you learned. And then 3 is spending some time each day quietly inside your own head, thinking about what you read, think about what you talked about, think about what had come up, what you've spoken about, and whether it's on a walk, on a run, or in the shower, or lying in your bed, but disconnecting from all the tech we have and being comfortable in your head, I think will set you up well to be successful as a scientist going forward.
Yeah, that's a good point. And I feel like now with all the technology, it's hard to disconnect. You're always it's very hard to be bored and sit with your mind, like you had said, and that's a critical part of being human. I think just being by yourself and really thinking about stuff you did before, or maybe just questions that you have. Yeah,
and I agree very much so. And maybe to the very first point, I think also that the value of content has been diminished. And I think it's so important to store the content in your own head. Yes, these machines are remarkable on the ability to search so quickly. But I think that some of the biggest innovations, ideas come from making connections between apparently seemingly disparate bodies of knowledge. And your brain does that in ways I don't think we fully understand. And if you don't have those bodies of knowledge stored, you're not going to make those connections. And so you can, know that how to get after body knowledge of A, body knowledge of B on your machine, but actually committing to having that and being that expert gives you the chance that you can make that connection that nobody's made before.
Yeah, sure. You just mentioned the do good science. Actually, that's a very good point. We heard first positive feedback from your previous postdoc students and talk about your mentorship. So tell us the do good science, what meant to you.
Yeah, I think it's really focusing on the work at hand. It's trying to find where there's a gap in knowledge or where there's maybe a real medical need that you need to fill, whether the gap or address the need. And it's focusing on the product, right? Humans love storytelling, they love gossip. Again, we have a way of getting so complicated. We have our busy, messy lives. And on top of what we're doing in the lab. And so it's focusing on the problem and really thinking about it for a long period of time. Are you on the right problem? And, digging in the literature to see, you'll be surprised to see how much has been covered.
Yeah.
But also how maybe that there are still opportunities. People think Elon Musk invented electric cars. That's not true. He's created a company that really is remarkable on electric cars. But if you go to the car museum here locally at Lars Anderson Park, you'll see at the turn of the last century, so in the early 1900s, we had electric cars. It was actually the first choice of tech. You open up, they've got this car where underneath the hood there's like 200 car batteries. They're all connected, and the car could go 200 miles. And so, the point here is digging the literature to see what's there and what was successful, what was not. But it's really focusing on the challenges and then also realizing that your good idea may not work.
Yeah.
And so the other point that I like to make on this do good science is that innovation doesn't occur just at the conception and the beginning of the project. You need to innovate every day at many stages of your project and be open to what your system is going to show you. So that it may not function the way you like, but you can build on it, or maybe there is another functionality that's being shown that you can leverage or exploit. So it's the do good science is to really also to avoid and forget about the other stuff, whether it's worrying about your grant or what happened to your paper, or did you not get the promotion, not get the award, or somebody mad at you. It's just, at the end of the day, what we really are in this for is to do good science, whether it's from the basic side or the applied side. And you can lose that in the midst of all the other things and all the distractions that we increasingly seem to have.
So during the interview for the citation, you were asked about the similar question, and I quote it here for our listener. It's like, as you find your field or your field of interest, commit to becoming an expert. Don't try to become an influencer. Right?
I like that's good advice from me. I think a lot of young folks are focusing too much on the online world, and I really value experts. And by an expert, it's somebody who's really mastered a body of literature, who's mastered a skill, mastered an area, and you don't become an expert because somebody said you're an expert. You became an expert because you mastered that content, you mastered that skill, that area. Nobody needs to even know you did it, but you did. And there's real value to that. And another advice I always give to young people is you should know what you know and know what you don't know, and be confident in both. Now, as an expert, you now know a lot more. And the influencers, I do think that this online fixation by many folks is not right. I was surprised by so many young people who were finding about new papers primarily from Twitter. And why would I want to be influenced so strongly by what other people are playing up? I should myself want to go in and just track the literature and make my own choice on which paper I'm going to read or what I'm going to follow versus some Joe Bag of Donuts who's become an influencer to come in. So I don't value the influencers. I don't think it should be a good goal. I think become an expert, master an area, and become somebody who's doing impactful work. And with that, maybe you become an influencer. But I think the folks who focus more on self-promotion and increasing the number of followers versus committing to really mastering an area, a set of content, set of skills, are losing out.
Yeah, and it goes into like technology, all the benefits and like drawbacks. It's like it's so easy to get information now, but there's so much more info you have to sift through and decide if it's good or not, which is kind of the issue with the influencer kind of model. You obviously talked a little bit about your science, mentorship of trainees, and a big part of your career has been entrepreneurship as well. And you've co-founded several biotech startups as well as a nonprofit called Phare Bio. Can you talk about or tell us a little bit more about how you got into entrepreneurship in biotech?
Yeah, I think it's interesting, from a historical perspective. So I started as a faculty in 1990. And in 1990, there were not very many professors doing translational work. And if you did, it was viewed a bit askance. You weren't taken as seriously as a scientist. You were viewed more as motivated by profit, maybe a bit greedier, maybe not as serious a scientist. That began to change in the late 90s as stock market was taking off and people were getting excited about how they could leverage their expertise to maybe make a buck on the side and get engaged in biotech. But what's interesting more so in the academic space is that the interest in impact has changed. So in 1990, the notion of impact was really about what papers did you publish? Did they themselves have an impact? Were they cited? Did you make a discovery? Did you have a breakthrough? Did you help launch a field? Who did you train? Now it's different. Now it's really how many patients' lives have you touched?
Yeah.
Do people use your technique? It's a very dramatic change over the last few years. So if you look at the highest levels in science, it's now that. And, this marvelous office, there's only so much I can do from behind here with our academic work and our trainees. And so you want to see if your stuff gets translated. So now you get engaged as an advisor, maybe a scientific co-founder with different companies. And, we've had some very nice success of things that have been moved into the clinic and as products, and some that hasn't worked out. I think in the biotech space, which right now is not doing well, broadly we're in a biotech winter of sorts, I think we need to re-engage the investment community, and with an eye toward long-term investment. In biotech, you need to be willing to fail a few times as you're advancing new tech, new platforms, because you're going to fail a few times before you get to success. And I think that was lost out in the last several years. So I think it's important. It's been meaningful. It can be frustrating, given, I think, the conflagration of competing interest from the founders, from the investors, from the workers, from the physicians. I think we need to do a better job of having those those interests align.
That's the reason why you have the social nonprofit, the startup called Phare Bio.
Yeah, so Phare Bio, Phare, it's actually French for lighthouse. And so the notion of lighthouse is a public good. We think of antibiotics as a public good. So Phare Bio is led by Akila Kosaraju, a marvelous CEO, visionary, and so positive. And it was set up really to take the most promising molecules from the antibiotics AI project and advance them towards the clinic through partnerships with the few existing biotech pharma still interested in antibiotics, through partnerships with nation states, other philanthropies, foundations, and big donors. And they're doing a marvelous job, primarily with support right now from ARPA-H. And we set it up primarily because the market for antibiotics is broken, in that antibiotics are sold quite cheaply. You typically take them acutely, yet it costs as much money to develop an antibiotic as it does a cancer drug or a blood pressure drug, which can be sold much more expensively, and you can take over many months, years, and the rest of your life. Similarly, very best antibiotics are often shelved to preserve them for when they're really needed after resistance arises. And so the business models for antibiotics companies are broken. And we set up Phare Bio with that in mind, and they've done really quite well. And I'm hopeful that they'll continue to play an important role at addressing the AMR crisis.
Yeah, that's a good point. It's like multifaceted with like the pharmaceutical role of it as well as the healthcare role.
That's right.
In the antibiotics.
Yeah.
Okay, so you have a lot of experience with entrepreneurship. When you're like having your trainees and advising them, I imagine a lot of people that join lab are interested in entrepreneurship. So how do you like kind of promote that, or like approach that situation, advising people in science as well as entrepreneurship?
Yeah, I'm not sure I promote it, but I'm supportive of it. And I tell the young folks here, because they're primarily coming from the science and the tech side, that I wish it was different, but our science, our cool tech is only about 5% of the story. That it's very much the business side, the market need, and then your business team. 1. 2 is to be very picky in who you team up with for your business partnership. That it's not like an academic collaboration that you could end tomorrow. A business partnership is more like a marriage.
Term sheet.
You're now in, and I think too often these young folks are teaming up with people they haven't done the due diligence on, and you want to make sure that you're aligned with your vision, your expectation, and really dig in. Are you compatible, and does it make sense? And I think young folks don't get enough advice in that regard. And then be careful. That for the most part, you're not trained as a grad student to be an entrepreneur. You're trained to do research, and so also know where your strength is and know what you don't know. And just because you came up with a really cool idea doesn't mean you're ready to be the CEO of a biotech company. It means you're really good at doing research. And so, think about how you can find folks that can complement your strengths to get to success.
It's a good point. Yeah, I imagine for a lot of scientists are very gifted in science, and it's tough for them to go to like a new area where like being a good scientist and knowing the science doesn't really translate to success and starting.
I think it's right. So I in the business world, I still don't understand market pulls and what drives markets. And I haven't studied it, but I have observed it without successful insight being gained. And I know that. And so I appreciate the folks that have meaningful insight that's not just kind of everyday knowledge. An odd example would be if you ever listen to Bill Belichick when he appears on the Manning Brothers broadcast. These guys talk about football in a way that is a whole different level, both in terms of language, but also insights of what they see that the typical viewer doesn't see. Find that person in the business world that can do that for you, and be patient to find that person. Okay, Belichick's maybe one of the best, easily one of the best coaches of all time. But can you find that person that has that differentiating insight that just a typical smart person doesn't have because they lack the experience?
Yeah. Okay.
That's good advice.
Yeah, I think that's you're in the synthetic biology. That's the field is also the question is about ethic. Because in 2010, you were appointed by President Obama as one of the member of the Presidential Commission for the study of bioethic issue. In the 2016, Craig Venter claimed his institute had synthesized the first minimal synthetic bacterial cell. Yeah, someone definitely criticized that. Tell us your thoughts about ethics of synthetic biology.
Yeah, it's interesting. I think as with any technology, new or old, there's dual use challenges. Meaning, you generally are advancing it with the good guys in mind. Can you improve human health? Can you improve planetary health? Can you address challenges? But there are bad actors out there also looking to exploit things, whether, for example, to make a bioweapon or to mess things up from a crop standpoint for a nation. And I think synthetic biology, unfortunately, increases the possibility that the bad guys can program biology to do nasty things. But it also opens up tremendous possibilities. On the case going back to Craig Venter, his team did this marvelous effort where they synthesized a natural genome and transplanted it into a related microbe. And then Obama's bioethics commission, which I got to present to, was my role, about synthetic biology. We're very open to it, and it was interesting. The non-scientists on that commission were really worried about gain-of-function mutations that would occur spontaneously, which I've not seen happen in my 25 years of doing synthetic biology. The biologists on the commission were worried about loss of function mutations that would disrupt the functionality of what we had designed, safety switch. And that happens all the time.
Oh.
And so the interesting challenge is, how do you make sure that what you engineered can function when you need it to function and doesn't get evolved away due to fitness challenges? Now, coming to the ethics, I think our field has done a good job of addressing social concerns and ethics from the very beginning. Maybe even more than we probably should be engaged, frankly, in that it seems to mix with scientific conferences, and I think we probably could have them a little better divided. But I think we've done generally a good job, and notably the conclusion of Obama's Bioethics Commission in 2010 was that we didn't need additional safeguards on synthetic biology, that the safeguards that were put in place with the Asilomar Conference in the 1970s were still sufficient to deal with what was being done in synthetic biology labs. And I think that probably a similar thing holds now. Again, that could change with increased ability to synthesize, increased ability to design with AI. But I think we just need to keep an eye on things.
Yeah, it's really good to hear. Obviously, synthetic biology has a lot of applications, but it's good to hear the scientists are very well in tune and aware of the ethics and plan accordingly.
Yeah.
So in the recent September issue of Scientific American, an expert from Iran actually discussed if and how AI can kill off humans. One possibility he discussed is that somehow evil AI release the pathogens to kill the humans. I think what you did actually opposite is to use AI to fight off this already existing pathogens. Right?
Yeah, no, that's right. So it's interesting. Pre-pandemic, I remember having a number of discussions with national leaders, primarily from the Department of Defense, CIA, DIA, pre-pandemic, and there was a lot of concern about synthetic biology, and AI was not yet on it. But the idea of would we make something nasty, and I kept telling them, you keep bringing this up, and I'm much more worried about what's going to happen with nature. And that nature evolves interesting things, and the nasty things in my lab are natural schemes, and we should have more efforts focused on what's evolving naturally from a pathogenic standpoint that's going to come get us. Now, again, we're not sure what the actual origins were of COVID, SARS-CoV-2. We'll leave that aside. But, the next outbreak, the next pandemic is coming from nature sometime soon, and I think we're ill-prepared. Will there also be concern on bioterror? Possibly. There have been small little instances in the last 30 years, but much greater death from naturally
Yeah.
evolved systems, and I think we need greater efforts. Similarly, then, from whether AI could design or human could design it, boy, nature right now is so much cleverer than we are.
Yeah.
And I think we need to be better prepared for the next outbreak, for the next pandemic, of course. And it's interesting of how our memories are short, and there doesn't seem to be much discussion at all about how to get ready for the next pandemic.
Yeah, so really only like 5 years ago, and it's like
5 years ago when it started. It's not even, we're only a few years out.
Yeah, and it hasn't really ended too. The fact that we're still.
So I would say the pandemic has ended, but that SARS-CoV-2 is with us, and it will be seasonal background, and you have to take appropriate precautions. I don't think we're going to eradicate that bug.
Yeah, I agree. And we kind of talked a little bit about use of AI for identifying new antibiotics, potentially antivirals, and there's a lot of buzz about what AI could do. So we're wondering if you could maybe just share a little bit about what you're most excited about in terms of what AI could do, and what do you believe it actually can do, and what do you think people are thinking it actually can't do?
I think AI is a useful tool, a tool that's growing in use. Nothing magical about it. I think people misunderstand it. I think it's increasingly being used in biology and medicine and biotech and drug discovery, but it's still early days, primarily because we lack the data sets to properly train. We lack the multilingual research groups to advance it. I like to say that really for the next decade or so, at the interface of AI, biology, medicine, the most successful groups will be the ones that handle three different things. One is they generate their own data to train and to validate. They have multilingual groups of young people across multiple disciplines that can talk to each other, and they're really effective at integrating human intelligence with machine intelligence. And that the complexity of biology, complexity of chemistry is such that AI is going to give us a bit of an advantage, but we still need to be able to ask those questions, merge with our human intuitions and insights from the experts, whether medicinal chemistry or microbiology, to really get after what matters. I think, again, it's early. The AI is only as good as the data you put in. I think, again, people have misunderstandings. ChatGPT, at least early versions, was not designed as an information source. It was designed to generate text. But many of my colleagues were using it as an information source. Let me check ChatGPT. And I gave them I said, well, you're asking it stuff you don't know, and it produces this thing that sounds reasonable. I said, ask it detailed questions for which you're an expert, and you'll be really disappointed in its answers.
Yeah.
And thus, if you're disappointed in the answers for which you're expert, why are you so confident and accepting of the answers in the areas you don't know, given that it's likely a similar level of inaccuracy and hallucination? And so, again, I think that even very smart people don't really understand what underlies these technologies. AI's been around since before I was born, and we've had different waves, and I've lived through them all and been familiar with them. This is interesting. I think it's really ChatGPT is what got people quite excited. It's something that can be used so easily, earlier DALL-E. But ChatGPT, where we're increasing on our machines, and now it's this system that can interface with us through text and language. And again, seems magical in how it goes and becomes a thought partner, if not replaces our thinking. And the latter is the danger part, if it's replacing your thinking and ability to express yourself. So I'm both encouraged and a little discouraged with how humans are using the AI.
Great.
Yeah, no, I agree. Yeah, it's like there's like a lot of hype, and it's hype in certain areas, and maybe it's being pushed into areas that probably people shouldn't be as excited about.
Yeah.
Yep. Wrapping up our episode. So, I will ask question. What's your hobby?
Yeah, I like to read books. I like to walk. I like to talk with friends and family. I used to be a runner, so I would say, but my body broke down enough, knees, back, calves, that I don't run. But I walk. So I still walk many, many miles each day by myself and with family members, friends, and I really cherish that time.
Yeah, and we're in your office right now. It's kind of like a library, and you have a bunch of books. Would you have like a favorite one that comes to mind?
Yeah.
I wouldn't say I have a favorite one, but many favorite ones. I think back to my undergrad days. I would say there it was both Richard Rhodes' Making of the Atomic Bomb and Douglas Hofstadter's Gödel Escherbach. As a grad student, it was a combination of Richard Feynman's Surely You're Joking, Mr. Feynman, Enigma, that was about Alan Turing, Chaos by James Gleick, then early faculty It was probably Robert Kanigals, the man who knew infinity, about Ramanujan. More recently, within my own space, in the antibiotic space, there's a book by Steve Johnson, The Ghost Map, about John Snow's efforts to really uncover the cholera outbreak in London in the 1800s was fantastic. So there's a lot of marvelous books up here. And again, I encourage people to stay, still read books. I think they're a marvelous source for inspiration, and they give you great historical context in the scientific world.
Okay. Is there any like a sci-fi movie or movie, you like?
The biggest impact was Rocky. So I remember as a kid going
Rocky.
Rocky. I mean, I very much like the underdog spirit, and I think in science you're always the underdog.
Yeah.
Against nature.
Yeah.
Nature's always got the upper hand, and I like being the underdog.
Yeah. It's really powerful. It's like Rocky, even though it's a sports movie, it like translates beyond that.
It does, in so many ways.
Yeah, and like we were talking a little bit before and after. It seems like you're a big sports fan, and you grew up initially in New York and then moved to Boston. So could you share a little bit about your allegiances between New York and Boston sports?
Yeah, so I was born in the Bronx. My dad had worked at Yankee Stadium. He grew up in the shadow of Yankee Stadium. But I then grew up in Queens, and as a kid was a fan of the Nets, Mets, and Jets. But we moved to New England, southern New Hampshire in 1975. I was entering 5th grade and became pretty quickly a sports fan, Boston sports fan, Red Sox, Patriots, Celtics, and remain now a sports fan. Married a Bostonian, so my wife was born and grew up and lived in Massachusetts. She's a rabid Boston sports. I'm not rabid, but I'm very supportive and very interested in sports. Both my kids, my daughter was a runner here at MIT. My son was a track athlete at Duke, and my wife was an All-American runner, as was my daughter, in college. And I ran at Holy Cross, and so it's been a big part. And it's interesting from a science perspective. I think young students today, the good students aren't allowed to fail at a level that they should. And I think you need to know how to fail if you're going to be a successful scientist. And sports allows you to fail. No matter how good you are, you're going to fail. Meaning you're going to miss your basket, you're going to miss a shot, you're going to strike, you're going to let the goal in, you're going to miss the goal. And so no matter how good, and learning to deal with that is really valuable. And I think my experience as an athlete shaped me as a scientist in unexpected and very important ways.
That's a good point. I think there's failure in everything, but in sports, it's like very visible. You see the athletes
every day, every practice,
day.
whether by yourself, in front of the fans and your team, you're going to fail. You're going to fail on a regular basis. Now, if you're really good, you're going to fail less than the other guy, but you're still going to fail.
Yeah, I think that's a very interesting point. Talking about how to dealing with the failure. Actually, we also talked about that before in the interview, the failure, because that's like what we have been facing every day for scientists, right? If it doesn't work, something doesn't work. But here, as a question is like any time, the moment, like, okay, maybe you should give up. Something like you should not do it. You're not good at it. It's not your like fit.
Well, so a few points, right? So the phrase on failure. One of my favorite phrases is the successful scientist is the person who can go from failure to failure with undiminished enthusiasm. So I think that as you're a failure in terms of say giving up on a career pursuit, there's this American adage, whatever about quit. You shouldn't quit. You should quit if, in fact, you're in a really bad situation and you don't want it. You shouldn't quit if you want it. And that's where the important distinction is really made in the American adage. Now, in terms of a project, it's one of the more difficult decisions as a lab director is when you need to move on from a product. Some products don't work, and you just might be in the wrong direction. So from an engineering standpoint, I like to say to my team, if you give me zero signal, I can't do anything with zero signal. You give me a little bit of signal, I can optimize, I can enhance that signal. You can then see, can you get enough where there's enough functionality? And so it's really making those decisions based on what's happening in front of you right now for your project, what's happening in the outside world. Are you being scooped? Has the world moved on? And it's the most difficult decision for a young person to make, which is putting in the garbage can what might be 80 hours or 80 weeks of work that you can't salvage or kind of get to the end. That's very difficult, but sometimes you have to do it. There may be ways you can make lemonade out of lemons that sort of thing. And okay, I don't want to spend too much time on that because you should move on to the more exciting, potentially successful project. But yeah, it's a big challenge, and it's hard to kind of assess that. And it's where, again, I always encourage all my young folks, be open to what your experiments are showing you. You go and, here's my hypothesis, I think, but boy, see what else, because those other things may be the things you could build upon for your next direction, or maybe the more important path for the project. Mm-hmm. But sometimes, you need to move on from a project.
Yeah, this is all good advice for science and outside of science, so it's a perfect way to end our interview. But we want to say thank you so much, Jim, for taking the time during your day to be on our podcast.
Yeah, so
I enjoyed talking with you both.
You have just listened to Harvard Griffin, Jesus, Biotech Club's The Pulse Podcast with your co-hosts Fang Xie and Yosef Baidi. Subscribe to our podcast on Apple Podcasts, Spotify, YouTube, and Substack, and catch our next episode. For more resources, please head over to our website, www.thepulsepodcast.org. Thanks for listening.